Hello again and welcome back. In this part 2, we're going to look at the role of plasma membrane proteins. And by doing that, we'll look at their functions as well. By looking at their functions, we're going to look at quite a lot of the processes that are vital in terms of cells, because of course they don't exist as separate entities. They have to have communication with other cells, with the rest of the body, with the circulation. So those plasma membrane proteins start to become very important, and the processes that they engage with. So that'll be this part. So plasma membrane proteins then, they can be integral. So in other words, they extend into the lipids of the membrane. They can be transmembrane, which means they go the full width all the way through, or they can insert into the membrane itself partially. Peripheral proteins are generally intracellular, and they're associated with, or sometimes bound to, integral plasma membrane proteins. But if you remember the structure of the plasma membrane and what it's made of and the implications of that, it has phosphate groups. Those phosphate groups are in contact on either side of the membrane with either the extracellular fluid or the cytosol within the cell, both watery substances. Therefore, the outer and the inner surface of the plasma membrane is hydrophilic. It likes water. However, of course, in the middle between those phosphates, because we've got a phospholipid bilayer, we have this lipid center with the lipid tails hanging into the middle. Therefore, the center of the plasma membrane is hydrophobic. It dislikes water. If you've ever put a greasy pan in a bowl of water without too much detergent, then you'll notice what happens. The lipid separates out. It doesn't like water. It's not soluble in water. So we have a hydrophilic outside and inside, and in the middle of those two areas, we've got a hydrophobic area. So what does all that mean in terms of our plasma membrane proteins? Well, integral plasma membrane proteins, whether they be transmembrane or partially through the membrane, have to have different zones. They have zones that are hydrophobic, or the opposite, lipophilic, they like lipid, or they have hydrophilic zones that like the water. So those hydrophilic zones, of course, are going to be in contact with the extracellular fluid or the cytosol. Peripheral proteins are generally hydrophilic because they're going to float around in the cytosol primarily. But they might have small hydrophobic regions or little anchors in case they need to embed in the membrane. So what about their functions? Well, we've got transport for a start off, and this is logical, isn't it? We've got a plasma membrane. We need to get solutes through that plasma membrane, either into the cell or out of the cell. And that transport has to be regulated. We can't let everything through willy-nilly, because thereby lies the path to chaos in terms of the cell. So we have permeability in terms of what and when. We'll talk about those again in a minute. In terms of the structures involved in transport, we may not have any structures. Again, we'll talk about that in a minute. But we might have ion channels. We might have things called transporters, otherwise known as carriers. We might have pumps, and we might have water channels, aquaporins. Signaling is another main function of those plasma membrane proteins. So here we're talking about detecting extracellular messages or creating intracellular messages as a result of something binding extracellularly, or indicating cell characteristics. So here our structures might be receptors, markers, or even enzymes involved in signaling. Intercellular interaction with adjacent cells. If you think about it, it's no point in having a cell not attached to anything. It's got to be attached either to adjacent cells or to the extracellular matrix to hold it into the form of tissues. Tissues then become organs or key structures. So we have to have links between adjacent cells, whether they be permanent, whether they be temporary. And this is by membrane junctions. And we'll talk about a few different types of junctions: tight junctions, adherence junctions, desmosomes, gap junctions, and also cell adhesion molecules. And finally, and this will be part 3, we'll talk about maintaining ionic and hydrostatic balance. If you remember from your pre-reading, between the intracellular and the extracellular compartments, we have chemical disequilibrium in that we have different chemicals in the different environments, so different ions. But we have a hydrostatic balance. We have an isotonic situation between the intracellular and the extracellular compartments, whereby we have net movement of water. And that's important to maintain. So this is where we're talking about the generation and the maintenance of a resting membrane potential. And we'll look at that in part 3 alongside sodium channels, potassium channels, and then we'll have some leak channels. And then in terms of maintenance, we'll have the sodium, potassium, ATPase pump. That will be part 3. So transport then. Obviously not across the membrane by a car. We have different types of transport. And there's important differences really. Passive transport doesn't require energy. Energy from our usual source, ATP that is. So no ATP required because it doesn't require energy. What does happen is that transport, whether it be ions or whether it be other molecules, in and out of the cell is mediated by gradients. If you remember, we mentioned an electrochemical gradient before when we were talking about the protons in the intermembrane space. Electrical gradients. Electrical gradients exist because opposite charges of any charged solute, whether it be an ion, in other words, a charged atom, or a molecule, attract each other. So positive is attracted to negative, negative is attracted to positive, like those magnets that come together if you put the opposite poles together. That creates an electrical gradient. Chemical or concentration gradients are about individual solutes. So this is regardless as to whether they're charged or not. Even if we're talking about ions, we're talking about the chemical itself, the element itself, not the charge it's carrying. That's separate. So we're talking about individual chemicals wanting to move from an area of higher concentration to an area of lower concentration to equalize out. So this is going to be specific to a chemical. For example, glucose will follow a chemical gradient and move via passive transport into cells, because when you eat a Mars bar, there's more outside the cell than there is inside the cell, because of course you used it for glycolysis. So it applies to any chemical. They will move or want to move, if they're allowed to, from an area of higher concentration to an area of lower concentration. Osmotic gradients are about the movement of water. So here we haven't got the solutes moving, we've got the water moving. So water moves to an area of higher osmolarity, and we talked about this in the module preparation. Think of it as water trying to dilute a glass of squash that you've made too strong. Pressure gradients we won't really come back to until we talk about the respiratory system. But individual gases diffuse from an area of higher concentration or pressure to an area of lower concentration or pressure. All of this is if movement is allowed, because we're going to talk about permeability in a minute. If the movement is allowed, it will continue with every gradient until there is equalization. At the point where there's equalization, of course, you no longer have a gradient. That gradient no longer exists. What will happen is you will probably get net movement. So as one molecule of chemical moves from outside the cell into the cell, one will move out from the cell to the outside. So you'll get net movement, but you have no gradient. In passive transport, if the substance is small and lipid soluble, so it has to be hydrophobic or lipophilic, it can diffuse directly through the membrane without any plasma membrane proteins. This doesn't apply to charged solutes. Ions, because of their charge, are not lipid soluble. Therefore, they cannot diffuse directly through the membrane. We can call this simple or non-facilitated diffusion. Non-facilitated because we don't have any plasma membrane proteins helping it. The solutes, though, because this is passive transport, still have to follow a gradient. So examples that can diffuse, and they're going to diffuse along pressure gradients, are oxygen and carbon dioxide. They can both diffuse through the plasma membrane, following their pressure gradients. Steroid hormones can diffuse through. The reason they can diffuse through is because they're based in cholesterol. They're made of cholesterol. So, and of course, cholesterol is a lipid. Alcohol is another one that can diffuse through the membrane. In passive transport, if the substance isn't lipid soluble, so it's hydrophilic or ionically charged, it has to cross the membrane via facilitated diffusion. In other words, it needs some plasma membrane proteins to help it through. So these structures could be a channel. Those channels can be specific for one thing, or they can be non-specific, or even partially specific. So an example might be ion channels. This ion channel we've got here is specific for potassium. Or aquaporins. Aquaporins are water channels. They allow water through. Another means of facilitated diffusion via passive transport is a transporter or a carrier. And what happens here, it changes shape to move a specific substance through when that substance has bound to the transporter. And it's generally for things like larger solutes, large molecules. It includes things like glucose transporters, aka glut transporters, or amino acid transporters. And what happens is the substance binds and gradually gets moved through. So it's less like a channel, it is more like a sort of squeeze it through method. Gradients, though, we still need to follow gradients. This is still passive transport, still has to follow a gradient. So in terms of glucose transport, as I said about glucose, didn't I? If you increase your blood glucose, it will move into cells because, of course, you've used the glucose within the cells for glycolysis, made some ATP with it. So generally, it's going to be lower than your blood sugar. So passive transport via ion channels, and we'll come across lots of different ion channels. Channels transport ions such as sodium, potassium, chlorine, calcium, remember that double cation, across the plasma membrane. However, this is still driven by gradients. In this case, they could be electrical, because of course we've got ions, they carry a charge. Could be chemical, so it could be specific to the chemical rather than the charge, or a combination of both, an electrochemical gradient. An electrochemical gradient means that both the electrical gradient and the chemical gradient are both moving in the same direction. So here's our rather little problem, of course. So all very well having these gradients, and we can say things want to move through. Whether they can or not is down to permeability. So ion channels are selectively and differentially permeable. So let's talk about that. Selective permeability is about what? So it's about what the channel will allow to pass through. And it's about the specificity of channels for one ion. So they may be slightly more generally specific. You might have channels that are specific for any cation or any anion. But generally they have some specificity. And this is about their selective permeability. They determine what they're going to allow through. Differential permeability is about when. When are they going to allow the ions through? So ion channels can be closed or open. And that depends on gating mechanisms. They have different gating mechanisms that can close them or open them depending on the stimulus. Sometimes we will come across an example later in the module. They have an additional time-controlled gate to lock them when closed. So even if they're stimulated, they still won't open. So differentiality, differential permeability is about when. It's about channels opening and closing. Selective permeability is about what. It's about the channels being specific for one or a few ions. So let's talk about those gating mechanisms. Channels, ion channels, can be opened or closed by chemicals binding to them. So on the channel you'll have a receptor. That receptor will be specific for a ligand, i.e. something, a chemical that binds to that receptor. The receptor is specific. And these are chemically gated or ligand-gated channels. You can use either term. Ligand is a slightly shorter word to write. But when these chemicals bind, either that channel will open or it will close, depending on the setting, if you like, for that channel. So we've got a ligand has bound to the channel has opened, and in this case, our channel is specific for sodium. So sodium is influxing through that channel when it opens. Again, we're talking about passive transport, so it's obviously following either an electrical gradient, a chemical gradient, or an electrochemical gradient. The other thing that channels can be opened or closed by is the adjacent electrical charge. So these sorts of channels, voltage-gated channels, have a voltage sensor. That voltage sensor has a point at which, once the adjacent charge gets to a certain threshold, it will open or close that channel. So it's detected by a voltage sensor on the channel, and that can open or close that channel. In this case, this is a voltage-gated, again, sodium channel. In this case, what's happened is around the sensor, we've got an increase. The charge around that sensor has become more positive. It's reached a point of threshold, and that sensor has opened the channel. And again, sodium is influxing. Other gating mechanisms could be mechanically gated channels. These tend to be a lot simpler. If you imagine you have a mechanical event such as stretch or pressure, it can physically or mechanically change the shape of that channel to pull it open or squeeze it open or closed. So enough of the passive then. Let's get a bit more active. Active transport requires energy. We have already talked quite a bit about ATP. So we need ATP to provide energy, because here what we're needing to do is to move things like ions against a gradient. So we need some energy. The structures that do this are known as pumps. So channels were about passive transport. Pumps are about active transport. Pumps have built into them the enzyme ATPase in order to hydrolyze ATP. Note, remember ATP synthase made ATP, synthesized ATP. ATPase, without the synth, hydrolyzes ATP to release energy. So ATP hydrolysis breaks the high energy bond between the phosphates, usually, remember, between ADP and the final phosphate. There you'll see the little sign PI, that stands for inorganic phosphate, but just helps to identify it. So it breaks the bond usually between ADP and the final phosphate, and this hydrolysis of that bond provides the pump with energy in order to pump something against a gradient. And we can pump things like sodium, potassium, calcium, hydrogen, our good old protons, and chlorine. These can be transported against gradients by pumps using ATP energy. And if you think about it, it's quite logical, because we have channels, and those ions go through the ion channels following a gradient. It stands to reason at some point we might want to correct that, because we can't let it carry on and carry on and carry on. So we might want to correct it. When we need to correct it, there's a good chance that we're going to need to move those ions back again against gradients. So hence, at that point, we need a pump rather than a channel. We need ATP for energy. Pumps vary. We have pumps that pump one ion in one direction. This is a uniport pump. We have pumps that pump two ions in the same direction. That's known as a symport. And we have antiport pumps that pump two different ions in different directions. So one ion will go in one direction, and then it will move another ion in a different direction, both against the gradient. So remember, all pumps move ions against a gradient using ATP hydrolysis to provide energy. ATP is hydrolyzed by ATPase on the pump. ATP is hydrolyzed and it becomes ADP and a free phosphate. And this hydrolysis provides energy. In part 3, if you remember, when we looked at the functions of plasma membrane proteins, we talked about resting membrane potential, its generation and maintenance. So in part 3, we will look at this major antiport pump, the sodium potassium ATPase pump, which maintains or restores resting membrane potential. Bulk transport. So bulk transport, here we're talking about vesicles. And if you remember, I said in part 1 about all the membranes in the cell are all phospholipid bilayer. So we can form vesicles from any of the organelles that have membranes, and those vesicles can then merge and meld with the plasma membrane of the cell. And this is what happens in bulk transport. So bulk transport involves the restructuring of the plasma membrane to form or incorporate vesicles so that solutes can be transported in or out via those vesicles. So vesicles can be created from the plasma membrane during endocytosis. Endocytosis is the movement of solutes into the cell. So the solute moves through the plasma membrane. The plasma membrane invaginates, it folds up to form a little pocket around those solutes. Eventually, the plasma membrane starts to close up again, and we've nearly got a whole vesicle formed. Of course, the next step is we've formed a whole vesicle, the vesicle's moved away from the plasma membrane. So yes, that vesicle is made with a little bit of the plasma membrane. Within it, we have the solutes that we've taken into the cell. So this is bulk transport endocytosis. The other way is exocytosis. This is the movement of solutes out of the cell. And we have mentioned exocytosis. So here the vesicles become part of the plasma membrane. So here's our vesicle with our solute for release from the cell. Could be some of those proteins that we made earlier, came out of the Golgi apparatus all nicely processed, ready to roll into their vesicles. That vesicle is going to move up to the membrane from the inside of the cell this time, fuse with the membrane starts to part, and then eventually the membrane opens out, releasing that solute that we've synthesized and now secreted from the cell. There's a lot more complexity involved in the formation of the vesicles, the fusing to the plasma membrane, the opening of the plasma membrane, all sorts of things like snare proteins going on and all of that, but you don't really need to know about those. But we do need to know that endocytosis, we're moving solutes in and forming vesicles. Exocytosis, we're moving solutes out, and vesicles are then becoming part of the plasma membrane after they've released the solute. So exocytosis is out, endocytosis is in. Exocytosis could involve the excretion of waste or the secretion of those proteins that we carefully synthesized and processed. Those synthesized proteins may be chemical or structural. In some cases, the structural proteins, or even the chemical ones, if we're making membrane-bound enzymes, may not be released from the vesicle once it opens out onto the surface. And this is a very good way of expressing more channels, more receptors, specific things. Say we decide we need more receptors for a specific ligand to bind to, or more channels for an ion. We can synthesize those through protein synthesis. We can then stick them to the side of the vesicle walls. Then when the vesicle binds to the plasma membrane and the vesicle opens up, we don't release the structures. The structures are embedded in the membrane. They just become a part of the membrane with that structure in. So this is a way of expressing new receptors, channels, pumps, or other proteins on the surface of the cell. In the same way, through endocytosis, we can take them back in again. If we decide we don't need all those receptors anymore, whatever, then the cell can take those back in through endocytosis in exactly the same way, obviously the reverse. Unregulated exocytosis is called constitutive. This is sort of the activities of daily living, if you like. It happens all the time. There doesn't have to be a specific or a regulated signaling mechanism to stimulate it to happen. It's just one of those things. We talked above about excretion of waste, so that would be constitutive. Regulated exocytosis is something that we intentionally stimulate through signaling processes. So there will be a signal process, often that will cause the release or the influx of calcium within the cell. Calcium stimulates exocytosis by interacting with the cytoskeleton. So often an influx or a release of calcium within the cell will stimulate regulated exocytosis. We just need a signaling mechanism to release that calcium. Endocytosis, we've got a few different types. Phagocytosis we will come back to in immunology. Phagocytosis, phago stands for eating, of cells, cytosis. And it's mainly an immune process, although it can be used with dying cells, so as a sort of clean-up operation generally. So it's a process where phagocytic cells that are capable of phagocytosing, phagocytes, will endocytose and break down pathogens, dying or infected cells, or damaged proteins. Pinocytosis is cell drinking. Always makes me laugh, that one. Pinocytosis, drinking. So cells endocytose extracellular fluid and the solutes within it. Receptor-mediated endocytosis is a bit of a misnomer, really, because even phagocytosis is receptor-mediated. But generally what we mean by this is where you get the formation of these protein coatings around pits within the membrane. And then the vesicle encloses with its coating on it and then has to uncoat. We'll look at an example in a minute. So phagocytosis then. Phagocytosis, as I said, this is actually receptor-mediated, really. A pathogen, dying cell, damaged protein binds in different ways that we'll talk about in immunology to a phagocyte. That phagocyte then puts up arms around it. They're called pseudopodia, fake feet, to engulf both the receptors and the target. The target is endocytosed into a phagosome. This is just another type of vesicle. That phagosome then fuses with a lysosome. A lysosome is a bit like the stomach of a cell. It's got full of toxic chemicals, enzymes that break down proteins and different things. So it's a vesicle of toxic chemicals. That lysosome fuses with phagosome to become a phagolysosome, and the toxic chemicals break down the contents of the phagosome, the target, in other words. That target is then exocytosed. So we've got a combination here of endocytosis and exocytosis. So the little debris left over once the target's broken down is exocytosed. Receptor-mediated endocytosis then, or RME. Although, as we said, phagocytosis does involve receptors, receptor-mediated endocytosis as a term generally refers to endocytosis that involves the formation of these protein-coated pits. The endocytosis of LDL is a classic example. This involves pits coated with a protein called clathrin. So the target binds extracellularly to a receptor. A protein called adaptin, it's like little anchors, binds to the intracellular side of the receptor. This then recruits and binds clathrin, another protein. The clathrin coats the inside of the membrane. So when endocytosis fully occurs, what you get is a vesicle coated in clathrin. That vesicle contains both the receptors and the target. The vesicle uncoats, the receptors are recycled back to the plasma membrane, and the target is processed in whatever way is most appropriate for that solute, that substance. Many viruses also bind and are endocytosed this way. Viruses need to get into host cells in order to replicate. They cannot independently replicate. And in fact, hepatitis C actually uses LDL receptors to get into hepatocytes. Coronavirus is thought to enter via clathrin-coated pits when it binds to ACE2 receptors, angiotensin-converting enzyme 2 receptors.
Cholesterol circulates in the bloodstream and then enters cells by receptor-mediated endocytosis. Instead of circulating freely, cholesterol molecules are derivatized and packed inside low-density lipoprotein particles, or LDLs. A protein and phospholipid layer surrounds the cholesterol molecules. The protein portion is recognized by LDL receptors on the surface of cells. An adaptor molecule, called adaptin, binds to the tail of the LDL receptor that protrudes into the cytosol. Adaptin recruits clathrin molecules, which start coating the membrane. Assembly of the clathrin coat causes the membrane to bend and invaginate, forming a vesicle that buds off inside the cell, taking with it LDL receptors and the LDL particles bound to them. Once inside the cell, the vesicle uncoats and fuses with the endosome, the intracellular compartment that first receives all endocytosed material. The endosome has low internal pH, which causes the LDL receptors to release their cargo. Empty LDL receptors are recycled to the plasma membrane in vesicles that bud off from the endosome. Each LDL receptor makes a round trip from the plasma membrane to the endosome and back every 10 minutes. Meanwhile, the LDL particles need to be disassembled. The endosomal content is delivered to a lysosome, which contains hydrolytic enzymes that can digest the particles. Free cholesterol is liberated, together with amino acids and small peptides generated by digestion of LDL proteins. The cholesterol is then released into the cytosol to be used in the synthesis of new membranes.
So that was transport as the functions of plasma membrane proteins, which brings us on to signaling. Signaling involves a ligand binding to a specific receptor to cause a cellular reaction. Every cell has many different types of receptors for different ligands. Receptors are generally specific for one or two ligands. It's like that lock and key principle. The ligand has to fit the receptor. So the specificity of the receptor for its ligand is based on that principle. So most receptors are specific for only one or two ligands. A cell may have thousands of receptors, though, for each specific ligand. And we're not just talking about plasma membrane here. Many receptors are also found inside the cell. If you remember, we've already talked about a transcription factor binding to a specific sequence of codons on DNA. To do that, it needed a receptor. So there are also many intracellular receptors. Receptors are said to have affinity for its ligand. Affinity you could interpret as the receptor's attractiveness to the ligand. So if the receptor has a low affinity, the ligand is less likely to bind. But you could increase that likelihood if you increased the amount of ligand available. If the receptor has a high affinity for its ligand, the ligand is more likely to bind. But again, it works that if there are significantly low levels of ligand, then that's going to reduce the likelihood of binding. Both the number of receptors and the affinity of the receptors for their ligands can vary. And this is downregulation and upregulation of receptors. Signaling can also be categorized by distance. So juxtacrine involves direct contact of adjacent cells. So this might be something that we'll talk about when we talk about epithelial tissue, contact inhibition. Cells are butted up against each other, joined by their junctions, and that stops them from going into mitosis, stops them from proliferating. So that's a type of signaling. Or you could have gap junctions, and we'll talk about those later when we talk about specialist junctions. Gap junctions form little channels between cells. Autocrine is quite funky. This involves a cell synthesizing and secreting a ligand that then binds to a receptor on or in the same cell. So the chemical is not going to any other cell, it is being used on that cell that synthesized it. So an example of this that we'll talk about when we talk about immunology is interleukins. Interleukins secreted by and binding to T lymphocytes when they're activated. And this helps to stimulate the T lymphocytes and also make them differentiate. Or intracellular signal transduction. So cells are producing chemicals that are then binding to intracellular receptors. Paracrine involves signaling between adjacent or local cells. So when we talk about inflammation, most of those inflammatory mediators, the chemicals involved in inflammation, degrade very quickly. So they only tend to bind very locally, unless you produce massive amounts of them. Or it could relate to things like neurotransmitters. Neurotransmitters are exocytosed from the presynaptic membrane. They travel across a very small gap and then they bind on the postsynaptic membrane, very short distance. Endocrine is going to form one of our full topics. This is about hormone signaling. Hormonal signaling, generally we're talking about a chemical getting into the circulation and having to go all the way around the body before it can bind to its receptors, wherever they may be. So we're talking about signaling over longer distances. We've already really talked about the simplest form of signaling. This is when a ligand binds to a chemically gated, also known as a ligand-gated ion channel, to either open or close it. So remember this, we had our channel with its receptor on it. The channel is closed because there is no ligand bound. Here the ligand has bound and the channel has opened. So that's a really simple form of signaling. The ligand is doing the signaling. The Na plus, the sodium, is being transported. And it's important that you keep that differentiation in your mind. If you think about it, the ligand that's binding to the channel is not being transported anywhere. It's signaling. But that signaling is facilitating the transport of sodium. Chemically gated ion channels, one place you can find them is the synapses. So they're found in the postsynaptic membrane of the postsynaptic neuron. The chemical in this case will be a neurotransmitter such as acetylcholine or GABA. So our neurotransmitter is exocytosed from the presynaptic neuron into the synaptic cleft. It diffuses across and it binds to chemically gated ion channels. Slightly more complex, signaling often involves second messengers, and we'll talk more about this when we talk about hormones. What's happening is a ligand binds to a receptor. Now that ligand can't get into the cell, it has to bind to the extracellular side of a receptor. When it does, it activates a peripheral protein that is intracellular. This is the second messenger inside the cell, just inside the plasma membrane. The classic example, this second messenger, is a G protein. It's called a G protein because it uses guanosine triphosphate, GTP, remember we made some in cellular respiration, for energy. The receptor is known as a G protein coupled receptor, GPCR, and you may see that in texts or in scientific papers. GPCR just means it's a G protein coupled receptor. There's an awful lot of them. So here's our G protein coupled receptor in our plasma membrane. At the moment, nothing is bound. Our G protein is hanging about nearby in the cytosol. You'll notice our G protein has 3 subunits. It has alpha, beta, and gamma. Alpha, at the moment, at rest, when it's just kicking its heels, has GDP attached, guanosine diphosphate. Along comes our ligand and binds. The G protein now moves to the intracellular side of the receptor. The next thing that happens, GDP comes off the alpha subunit and GTP goes on. So the alpha subunit exchanges GDP, which is no use to it for energy, for something that is of use, GTP. It's like it's swapped an empty purse for a full purse. It's not going to use it yet though. It's just hanging on to that little full purse. It's its bus fare home, if you like. So we've switched GDP for GTP. Next thing, the whole G protein moves away from the receptor. The reason it needs to do this is so that additional G proteins can come up and bind to that receptor. As long as the ligand is bound, more G proteins will come up, bind, and be activated. Now, having moved away the whole thing from the receptor, the alpha subunit disassociates from beta and gamma. So the alpha subunit still has its GTP. If you think about it, it's a bit like, okay, 3 mates want to go out. Alpha has all the money. His mates beta and gamma didn't get any money from their mother, the G protein coupled receptor. So beta and gamma are left kicking their heels at the bus stop. So they're left hanging around for a while. They do have some functions, but not as important as our alpha subunit. Alpha subunit has a full purse, so he has the bus fare to get home again. Alpha subunit now goes off and can do lots of different things. He's an activated alpha subunit. So the alpha subunit can activate or inhibit other enzymes, or it can directly activate and open some channels. The GTP, its little purse of GTP, it hydrolyzes that to release energy, just like our pumps and ATP, to release energy to allow it to return to beta and gamma once it's performed its role. As long as the ligand is bound, more G proteins associate and are activated. Well, that was a quick run through signaling. We'll come across loads and loads of instances as we move through the module, which leaves us with the formation of cell junctions for this part. So we'll talk about the four main types. So working down from the top, we have tight junctions. Tight junctions are a bit of a misnomer, as we'll find out later on in the course. Below that, we have adherence junctions. Below that, you can see a desmosome. And at the bottom there, a hemidesmosome, half a desmosome. These half desmosomes, rather than linking two cells together, help to link cells to the extracellular matrix, basically collagen layers, to anchor the whole tissue in place. And also, we've already mentioned gap junctions. Gap junctions are like little tunnels between cells. Tight junctions have two main functions. One is the regulation of the passage of solutes and ions through the space between the cells. This is called the paracellular pathway, intercellular, if you like, between cells. And it can regulate what passes through there. This is why really the name tight junctions isn't that accurate, because it does allow some things to pass through. Tight junctions also block the movement of the plasma membrane proteins between the apical and the basolateral surfaces of the cell. So if you think about our endocytosis and exocytosis, for example, what we're doing is we're taking away bits of membrane, we're adding bits of membrane. The potential is for any channels or receptors that we want at the top of the cell, the apex, the potential is for them to start moving around the sides as we have more and more exocytosis. The tight junctions stop those plasma membrane proteins basically moving down the sides of the cell where we don't want them. Adherence junctions basically initiate the first cell-to-cell contact, and then they maintain that. So they hold cells together. Desmosomes have these plaques on the inner surface of the plasma membrane on both sides. So here we've got two plasma membranes of adjacent cells. Both have their green plaque there. And then we have filaments between the two cells, intercellular, between the adjacent plasma membranes. Those filaments can be unzipped. They look a bit like a zip. The other side of the plaque is joined to the cytoskeleton via, in this case, intermediate filaments of the cytoskeleton. This is the intracellular side. Desmosomes generally are quite good at resisting shearing forces and mechanical stress. If you think of those bridges that have got like the sort of zigzaggy lines, at either end that allow the bridge to move a little bit. Desmosomes are a bit like that. So they're really important somewhere like the myocardium, because the heart, of course, is expanding, contracting under a lot of stress. So they're quite important there for preventing any tearing, any damage. But as we said, they can be unzipped to allow for any cell motility, allow the cells to move around, or for growth of the tissue, for proliferation of other cells into that space. Hemidesmosomes, as we mentioned, help to secure cells to the extracellular matrix. So basically they're holding the tissue as a whole in place by holding all the individual cells to the extracellular matrix. Gap junctions, as we said, are like little tunnels. They're tubes, and they allow solutes and ions to pass directly cell to cell. They're formed by protein groups called connexons within the plasma membrane of each cell. So you can see there we have each of the two cells, top and bottom, have their own connexons. The connexons are made up of 6 connexin proteins. And they work, it always reminds me a bit of a camera shutter there, to open and close. So each connexon is a group of 6 connexins. Those connexins can close or open the tube between the two cells. And these, again, are very important in the myocardium because what they do, and also in smooth muscle, they allow ions to move quickly from myocyte to myocyte, from muscle cell to muscle cell, so that any contraction can occur simultaneously. It's called syncytially. So the whole thing contracts as one because the ions can flow from cell to cell. They don't have to come in from outside the cell. They don't have to have lots of mechanisms. They can just go cell to cell to cell to cell like a system of tunnels. When they're open, solutes such as ions and glucose can get through. Whereas, of course, they're too small to allow any of the organelles through. Cell adhesion molecules tend to be a little bit more temporary, or they can be more permanent. They're much simpler structures. Here we're talking about sort of a small group of proteins rather than a big complex structure. Cell adhesion molecules basically are plasma membrane proteins that bind to their counterpart on a different cell or the extracellular matrix. And it can be very temporary, a bit like Velcro, or it can be more permanent. And there's various different types. There's integrins, selectins, cadarins, depending on their structure and function. And our cell junctions that we talked about, adherence junctions, for example, have these cell adhesion molecules between each side, holding them together. So they're a component part of bigger junctions, but they can operate in their own right. Their functions include attaching the cell to the extracellular matrix or to another cell. They're also responsible for stabilizing synapses between neurons. So you've got two neurons in very close proximity, but of course we don't want them to drift apart. Otherwise, when we exocytose the neurotransmitters, they're not going to get to the postsynaptic membrane. Remember our chemical signaling of neurotransmitters. And they're going to dissipate off into the extracellular fluid. So we need to hold those two neurons close together. So they stabilize those. Another thing is facilitating cell migration through tissues. So cells often have to move through tissues, and they do this a bit like sort of slugs. They use their cytoskeleton to change shape and squeeze through other cells. But cell adhesion molecules allow them to do that. They allow them to sort of grab on and pull themselves through. And certainly, again, when we look at the immune system, we'll talk about cell adhesion molecules, because there they have a really important role in helping the leukocytes, the white cells, to get from the blood into the tissues through the endothelium of the vessels and into the tissues, where they can start acting to clear things up and kill things off. Well, that was part 2, where we talked about plasma membrane proteins. There are other plasma membrane proteins available, and this was quite a simplistic view. There's other proteins like CD proteins, cluster of differentiation proteins, that identify cells. There's antigenic markers. Some of them we will come across during the course of the module, others you don't really need to know about. So we've left those out. You'll be glad to know. Meanwhile, as after last time, keep your glossary up to date, keep your notes up to date, your own notes, your own diagrams. Try and teach someone else, even if you're doing it pretending you're telling someone in your head until you get your confidence going. Do all those things, because you need to start getting this, the majority of it, more or less committed to memory before moving on, because it's only going to accumulate. So you need to be in a situation where you can remember most of this before moving on to the next part. I'll see you at your live session, and then see you in part 3.